In its broadest sense, biology is the study of living things. It is, from an objective standpoint, the science of life. All living organisms, regardless of shape, size, or complexity, are subjects of this scientific discipline. Biology examines the organization of life, the functions and patterns that define organisms, and the principles that govern their growth and development. From microscopic cells to vast ecosystems, biology concerns itself with every level of life's intricacy. This wide scope has given rise to numerous specialized branches, including evolutionary biology, cellular biology, genetics, and developmental biology, each focused on particular aspects of living systems. The question then naturally arises, what do we mean by life? While there is no single universally accepted definition, most living organisms share certain fundamental characteristics. First is cellular organization. Every living organism is composed of one or more cells, which carry out the essential functions of life. Second is order. All living beings are highly organized, with bodies composed of diverse cells, each housing complex molecular machinery. Third is sensitivity. Living organisms respond to stimuli from their environment. A plant bending toward sunlight or your pupils adjusting in a dark room are familiar examples. Fourth is growth, development, and reproduction. Organisms grow and reproduce, passing hereditary material, genes, onto their offspring. Fifth is energy utilization. All organisms take in energy to fuel their many biological tasks. The energy powering your muscles, for instance, originates from the food you eat. Sixth is evolutionary adaptation. Through interaction with the environment and with one another, organisms develop traits that enhance survival. Lastly, there is homeostasis. Despite external changes, living organisms maintain relatively stable internal conditions, an ability crucial for continued life. This naturally leads us to the idea of hierarchical organization in biology. Life is not a random assortment of components, but is structured in a well-ordered hierarchy. This hierarchy spans 10 levels, beginning with atoms at the most basic level and rising to the level of ecology. From atoms, molecules are formed. These molecules come together to create cells, which are then arranged into tissues. Tissues make up organs, which, when grouped, form organ systems. These systems work within an organism. Organisms, in turn, form populations. Populations of different species living together comprise communities. And finally, these communities interacting with their physical surroundings constitute ecosystems. At the cellular level, atoms combine into molecules. These molecules form organelles, tiny functional structures within cells. Examples include the nucleus and mitochondria. The cell itself is the fundamental unit of life. Some organisms, such as bacteria, are composed of a single cell. Others, including plants, animals, fungi, and many algae, are multicellular. Moving up, we arrive at the organismal level. Here, cells group into tissues, collections of similar cells functioning together, like muscle or blood tissue. These tissues combine to form organs, such as the heart or brain, each comprising different tissues arranged for a specific function. Organs are then organized into organ systems. The nervous system, for instance, includes sensory organs, the brain, and the spinal cord. At the populational level, organisms of the same species living in the same area constitute a population. Multiple populations of different species coexisting in a shared environment form a community. When this community is combined with its physical environment, the result is an ecosystem, a full dynamic system of living and non-living interactions. As we ascend through these levels, new characteristics emerge that were not present at lower levels. These are called emergent properties. For example, a functioning cell demonstrates abilities not found in the molecules that make it up. Similarly, proteins exhibit traits not present in their individual atoms. Take diamond and coal, for instance, both composed of carbon, yet radically different in structure and properties. These emergent properties are a central concept in understanding the complexity of life. Two foundational ideas in biology deserve particular attention: hierarchical organization and emergent properties. They serve as cornerstones for understanding life's complexity and integration. A core theme in biology is the organization of life, and central to this is the cell theory. All living things, from the tiniest single-celled organism to the colossal kilometers-wide fungus Armillaria ostoyae, are composed of cells. The cell theory rests on two major conclusions. First, that all living things consist of cells, and second, that all cells arise from pre-existing cells. These ideas underpin our understanding of biological growth and reproduction. Life's continuity from one generation to the next is governed by the molecular basis of inheritance. Even the smallest cell, invisible to the naked eye, is astonishingly intricate, more complex than a computer. Each cell's characteristics are determined by genes, which reside in the cell's nucleus. Genes dictate a cell's shape, behavior, and energy use. They are segments of DNA, a molecule composed of two intertwined strands of nucleotides that form the iconic double helix. The faithful copying of this DNA during cell division ensures the transmission of genetic information. This is the essence of heredity. There is also a strong correlation between structure and function throughout biology. By studying a structure, we can infer what it does. By knowing what it does, we can predict its form. The anatomy of a bird's wing, for example, reveals its function. The wing's aerodynamic shape, the strong chest muscles that power flight, and lightweight skeletal adaptations all coalesce to enable a bird to fly efficiently. Biology also concerns itself with the diversity of life. Scientists have so far identified about 1.5 million species, including over 260,000 plants, nearly 50,000 vertebrates, and more than 750,000 insects. The true number may be far higher. Some estimates range up to 100 million species. This vast variety is organized into three domains: bacteria, archaea, and eukarya. Bacteria and archaea are prokaryotes, organisms with relatively simple cells lacking a nucleus. Eukarya, by contrast, includes all organisms with complex cells, such as animals, plants, fungi, and protists. Within eukarya are kingdoms: Animalia for animals, Plantae for plants, and others. Further classification continues through phylum, class, order, family, genus, and species. 8 levels in total. How did this extraordinary diversity come about? The answer, universally accepted among biologists, is evolution. Through gradual change over time, life diversifies, adapts, and branches into new forms. Despite this diversity, life also shows remarkable unity. All living organisms are believed to have descended from a simple cellular ancestor that lived approximately 2.5 billion years ago. Certain features of that ancestor remain preserved today. All life, for instance, stores genetic information in DNA. All eukaryotic cells possess nuclei with chromosomes. These shared characteristics, conserved through evolutionary time, are not easily altered, suggesting their fundamental importance in the biology of life. The theory of evolution has roots that stretch back to antiquity. Several classical Greek philosophers entertained the notion that life evolved gradually. However, the thinkers whose ideas most deeply shaped Western thought, Plato and his student Aristotle, held firm to a contrasting view. They maintained that species were permanent, perfectly crafted entities, unchanging through time. This notion of fixity was reinforced in Judeo-Christian traditions. The Old Testament's account of creation upheld the belief that each species was individually designed, and therefore immutable. By the 1700s, biology across Europe and America was heavily influenced by natural theology, a discipline that sought to understand the Creator's design by studying the natural world. In this context, adaptations in organisms were interpreted as deliberate features, each one tailored for a specific role in nature. One of natural theology's key aims was the classification of species, believed to unveil a divine hierarchy of life. During the 18th century, a Swedish physician and botanist named Carolus Linnaeus sought to bring order to the natural world for the greater glory of God. Linnaeus established taxonomy, the science of naming and classifying living organisms. He introduced the binomial naming system, assigning each species a two-part name composed of genus and species, a convention that remains in use today. Furthermore, Linnaeus created a hierarchical system, grouping similar species into genera, similar genera into families, and so forth. At the time, he saw these groupings as reflecting the Creator's organization of life, not evolutionary relationships. Ironically, his taxonomic framework would later become instrumental to Darwin's arguments for common descent. As the 18th century drew to a close and the 19th began, scientists turned their attention to fossils, those petrified remnants or impressions of ancient life. Debate flourished over geological formations and how the history of life might be inferred from them. Paleontology, the scientific study of fossils, emerged through the work of French anatomist Georges Cuvier. Studying strata in the Paris Bassin, Cuvier observed that each geological layer contained its own unique assemblage of fossils. Moreover, the deeper the layer, the less the flora and fauna resembled those of the present day. Cuvier was among the first to assert that extinction was a common event in Earth's history. From one stratum to the next, species would appear and vanish. Yet, despite his detailed observations, Cuvier rejected evolutionary explanations. He favored catastrophism, the view that the boundaries between strata were formed by regional disasters, such as floods or droughts, events that wiped out species which were then replaced by others, migrating from elsewhere. His world was shaped not by the slow hand of evolution, but by sudden, dramatic upheaval. In contrast, the geologist Charles Lyell, a contemporary of Darwin, rejected catastrophism. Building on James Hutton's earlier work, Lyell developed the theory of uniformitarianism. He argued that the same geological processes observed today, mountain formation, erosion, and the like, had been shaping Earth throughout its history and at roughly the same rates. This suggested not only that Earth was far older than the 6,000 years proposed by biblical accounts, but also that profound change could result from slow, continuous processes. Darwin drew two important conclusions from Lyell's work. First, if geological transformations occurred gradually, then Earth's age must be vast, allowing sufficient time for biological evolution to unfold. Second, these slow processes, if acting consistently over long periods, could produce significant changes. Darwin was not the first to connect gradualism with biological evolution. By the late 18th century, several naturalists had proposed that life had evolved alongside the changing Earth. One of them, Jean-Baptiste Lamarck, put forth a comprehensive theory in 1809, the very year of Darwin's birth. Lamarck believed that life evolved from simple to complex forms and sought to explain how specific adaptations emerged. Lamarck's theory rested on two main ideas. The first was the principle of use and disuse. Body parts that were used extensively would grow stronger, while those left idle would weaken. He offered the example of a blacksmith developing a muscular arm, or a giraffe stretching its neck to reach high branches. The second idea was the inheritance of acquired characteristics, the belief that traits acquired during an organism's life could be passed to its offspring. The giraffe's elongated neck, Lamarck argued, was the accumulated result of many generations stretching higher and higher. While imaginative, this model was flawed. There is no evidence that acquired traits can be inherited. Charles Darwin, 1809 to 1882, on the other hand, not only established the theory of evolution on firmer ground, but also identified its driving mechanism. From a young age, Darwin was fascinated by nature. At 22, he embarked on a 5-year voyage aboard HMS Beagle, beginning in December 1831. His travels took him across continents and islands, offering him a broad survey of Earth's biodiversity. He examined birds, plants, fossils, and countless other specimens, making careful observations that would later reshape biological thought. Upon his return, Darwin spent years reviewing his notes and evidence. Eventually, he proposed that the diversity of life on Earth was a product of evolution through natural selection. In this view, new species arise from ancestral ones by the gradual accumulation of adaptations suited to different environments. Natural selection, Darwin argued, is the mechanism by which certain traits become more common in a population. Organisms better adapted to their surroundings are more likely to survive and reproduce. Darwin presented these ideas, along with substantial supporting evidence, in his seminal work, On the Origin of Species, published in 1859. The book was widely read and profoundly influential. It transformed evolutionary theory from a speculative concept into a central, unifying principle of biology. To understand the general meaning of evolution, one must first consider 5 central concepts: descent with modification, variation, natural selection, adaptation, and the origin of species. Darwin began with the notion of unity in life, observing that all organisms are connected through descent from a common ancestor, some unknown prototype that existed in the distant past. As these descendants spread into diverse environments over millions of years, they underwent numerous changes. These changes, or adaptations, helped them to survive and thrive in their respective habitats. This process, in which observable traits and genetic information are passed from one generation to the next, is known as descent with modification. It forms the bedrock of Darwin's view of life. The principle refers specifically to the transmission of heritable traits, those grounded in genetic material. To clarify, not every trait or feature observed in an ancestor is passed down. Only characteristics that are encoded in DNA can be inherited. For example, in humans, eye color, skin pigmentation, blood type, and a predisposition toward baldness are examples of traits that can be passed from parent to offspring. These are governed by genetic material. In contrast, traits acquired during an individual's lifetime, such as a blacksmith's enlarged biceps or damage to an organ caused by injury, are not encoded in DNA and therefore cannot be transmitted genetically. Heritable traits are passed through DNA, the molecule that holds genes. As changes occur in these inheritable traits, whether through mutation or recombination, new features can arise and gradually become more common within a population. This is how evolution unfolds at the population level. The second concept is variation. This refers to the observable differences that exist within a population. If we simply look around us, we can easily identify variations. Differences in skin tone, facial features, height in humans, variations in coat color among domesticated animals, or differences in the size and hue of flowers within the same species. These visible traits are often linked to what are referred to as dominant genes. The genetic material behind these traits forms an organism's genetic composition. By observing the visible traits, the phenotype, we can make inferences about the underlying genetic differences, or genotype, within a population. Over time, the genetic composition of a population changes. When these changes accumulate to a degree that they alter the nature of the population, evolution is said to have occurred. So what causes this variation? It arises from several genetic mechanisms, including mutations, which are changes in DNA, genetic recombination during mating, and gene flow, which is the exchange of genes between populations through migration. These changes introduce new genetic material into the population and diversify the gene pool. Variation is essential to evolution. Without it, there would be no material for natural selection to act upon. In short, evolution depends on the existence of variation within a population. And how exactly does evolution proceed through such variation? That brings us to the next key concept: natural selection. Natural selection, the third pillar in understanding evolution, can be considered from three angles: its definition, illustrative examples, and the different forms it takes. To begin with the definition, natural selection refers to the process by which organisms that possess traits better suited to their environments have a higher chance of surviving and reproducing than others of the same species. These advantageous traits, being heritable, are more likely to be passed on to future generations. Over time, the genes associated with these traits become more common in the population. In this way, nature selects the traits that best equip organisms to thrive under specific environmental conditions. A well-known example of natural selection is drawn from the period of the Industrial Revolution. As coal-fired factories spread across the landscape, thick smoke darkened the surrounding forests. In these soot-stained environments lived two varieties of moth, one white, the other black. On darkened tree bark, the black moths were better camouflaged, escaping the notice of predatory birds. White moths, by contrast, stood out starkly and were more frequently eaten. As a result, the population gradually shifted from a mix of black and white moths to predominantly black moths. This simple but powerful example demonstrates how environmental change can influence the survival and reproductive success of organisms, leading to evolutionary change through natural selection. To understand how natural selection operates, consider a hypothetical population of beetles. The first requirement is variation, differences in physical traits, such as color. Suppose the beetles come in green and brown varieties. The second requirement is differential reproductive success. Not all individuals reproduce equally, and various factors limit population growth. In this case, green beetles are more visible to predators and are eaten more frequently, reducing their reproductive success compared to brown beetles. The third requirement is that these traits are heritable. Brown beetles tend to produce brown offspring, and green beetles produce green offspring because these color traits are encoded in their genes. Given these three conditions, variation, unequal reproductive success, and heritability, natural selection occurs. Over time, the brown beetles, being better camouflaged and more likely to survive, become more numerous. The population evolves as these traits spread. If these criteria are met in any population, evolution through natural selection is not just possible, it is inevitable. Natural selection also comes in several varieties. Directional selection occurs when one extreme of a trait becomes favored, shifting the population in that direction. Disruptive selection favors individuals at both extremes of a trait, often leading to two distinct forms within a population. Stabilizing selection favors the average traits, reducing variation and maintaining the status quo. Sexual selection involves traits that increase an individual's chances of mating, even if those traits are not necessarily beneficial for survival. Lastly, artificial selection is the result of human intervention, where certain traits are deliberately chosen and propagated in domesticated species. When considering the consequences of evolution via natural selection, it becomes apparent that while the effects can influence both structural features and behavioral tendencies in organisms, the most immediately observable outcomes tend to be behavioral and physical adaptations. For this reason, it is worth examining adaptation more closely. Adaptation involves three main aspects: its definition, its mechanisms, and its consequences. To begin, adaptation is the process by which organisms become better suited to their habitats. In other words, it is how traits that improve survival and reproduction in a particular environment are favored and become more common over time. Adaptedness, on the other hand, refers to the state of being well suited to one's environment. The degree to which an organism can survive and reproduce under specific ecological conditions. Organisms must constantly contend with limited resources and competition for survival. Those with traits that confer an advantage in their particular environment are naturally selected, leading to a greater likelihood of these traits being passed to the next generation. The result is an ongoing refinement of fit between organisms and their environments. The consequences of adaptation are practical and direct. Traits that improve the odds of survival and reproduction enhance the persistence of those organisms in their respective ecosystems. On the other hand, organisms that lack such adaptive traits face a greater likelihood of reduced reproductive success or outright extinction. In this way, adaptation contributes to the growth and stability of populations by ensuring that favorable traits are retained and propagated. Turning now to the origin of life, the sixth point in our discussion, it is broadly accepted among scientists that the universe itself came into being roughly 13.7 billion years ago, and that the Earth formed about 4.5 billion years ago. The precise timing of life's origin remains unknown, and there is as yet no definitive evidence pinpointing the moment it began. In the early stages of Earth's formation, the planet's surface was composed of molten material. As temperatures cooled over time, conditions gradually stabilized. Earth's distance from the Sun, along with the presence of abundant water, created a hospitable environment for life to emerge. By contrast, planets such as Mercury and Venus are too close to the Sun, while Mars and Jupiter lie too far away, making them less favorable for the development of life as we know it. Life as we understand it is based on carbon, a versatile element believed to have been introduced into Earth's environment, partly through volcanic eruptions. Carbon, in combination with elements such as hydrogen, oxygen, and nitrogen, forms amino acids, the building blocks of proteins. Proteins, in turn, are essential for cellular processes. Enzymes and other catalytic proteins enable organisms to perform fundamental biological functions. The progression of life seems to follow a pattern, from simpler to more complex forms. Even if life began as small and limited in number, it expanded and diversified significantly over time. Importantly, any organism that relies on another for survival, such as a virus, cannot be considered a truly original life form. Viruses and similar entities likely appeared after the first autonomous organisms had already evolved. The earliest physical evidence of life points to the spontaneous formation of amino acids through chemical reactions in the oceans and lakes of ancient Earth, an event estimated to have occurred around 3 billion years ago. Some scientists, however, propose an alternative theory, that life may have originated on an exoplanet and arrived on Earth by some cosmic means. There is also evidence to suggest that RNA molecules, crucial for genetic information storage and transfer, could have formed through chemical reactions in Earth's early environment. This has led some researchers to argue that the defining moment in the origin of life is the emergence of molecules capable of self-replication and information transmission. Today, scientists continue to investigate these questions through laboratory experiments. Their goal is to recreate conditions that might allow for the spontaneous generation of life, or at least the molecular precursors to it. These efforts are part of an ongoing attempt to understand not just how life functions, but how it first began. Let us now turn our attention to the foundational theories that govern our understanding of living organisms at their most basic levels, cellular and genetic. In this discussion, we begin with the nature of cells, their types, internal components known as organelles, and the intriguing topic of cell cloning. The story begins in 1665 with Robert Hooke, a British scientist who first observed the cell. Using a compound microscope to examine thin slices of bottle cork, he noticed a multitude of tiny pores, which he described as cells, a term that evoked the small rooms in which monks resided. These observations marked the beginning of cell biology, though Hooke could not discern the intricate nature, structure, or function of these pores. Further development of cell theory would not emerge until the 1830s, when German botanist Matthias Schleiden recognized that all plants are composed of cells, and Theodor Schwann, a veterinary scientist, concluded that all organisms are made up of cells. These findings gradually propelled the field of biology forward. A cell is defined as the basic unit of life, encapsulating all the essential characteristics required for living function. However, it is not the basic unit of matter, as cells themselves are composed of a variety of molecules and atoms. Cells have the unique ability to reproduce, to respond to environmental stimuli, to inhale and exhale, to convert nutrients into usable energy, to expel waste, to maintain homeostasis, and to grow and reproduce. According to the cell theory, all living organisms are composed of cells, and new cells arise only from the division of pre-existing cells. There are two primary types of cells, prokaryotic and eukaryotic. Prokaryotic cells are generally smaller and simpler in structure compared to their eukaryotic counterparts. They lack a nucleus, possess fewer chromosomes, and are typically unicellular. Prokaryotes fall into two categories, archaebacteria and bacteria. Bacteria are among the earliest organisms to have appeared on Earth and can be found thriving in environments as diverse as land, sea, ponds, salt water, and even acidic pools. These organisms come in three primary shapes: spherical, rod-shaped, and spiral. Archaebacteria, meanwhile, are found in extreme environments such as volcanic springs, salty waters, and deoxygenated swamps. It wasn't until around 1990 that archaebacteria were formally recognized as living organisms. Eukaryotic cells, by contrast, contain a nucleus enclosed by a membrane and are composed of a variety of specialized organelles. They are further divided into two categories: plant cells and animal cells. Plant cells make up structures such as leaves, roots, flowers, and stems. Animal cells constitute components of the physical body, including the skeleton, muscles, blood, skin, and nerves. The two types of cells differ in several ways. Plant cells have cell walls and contain chlorophyll. Animal cells do not. In terms of shape, plant cells tend to be rectangular, while animal cells are more spherical, though shape is not the defining difference. Functionally, plant cells generate their nutrients through photosynthesis, relying on sunlight. Animal cells lack this ability and must obtain nutrients by consuming other organisms. For instance, human cells cannot synthesize glucose and therefore depend on foods like wheat, barley, and beans for nourishment. Though cells are too small to be seen with the naked eye, they are composed of smaller structures known as organelles, each with specific forms and functions. Among the most critical organelles are the nucleus, mitochondria, lysosomes, and the cytoskeleton. At the center of the cell lies the nucleus, which contains chromosomes, DNA, and genes. These genetic materials are structured hierarchically. Genes form DNA, which spirals into chromosomes. Their function is to encode the instructions required for cellular and bodily operations. During protein synthesis, messenger RNA, or mRNA, transcribes information from DNA located in the nucleus. This mRNA then passes through nuclear pores to relay its information to ribosomal RNA, which constructs proteins. This first stage is called transcription, and the second stage, during which proteins are assembled based on the mRNA's instructions, is known as translation. Mitochondria are somewhat unusual in that they contain their own DNA and genes, allowing them to carry out transcription and reproduce independently. While only eukaryotic cells contain mitochondria, prokaryotes are nevertheless capable of producing energy. Interestingly, although both parents possess mitochondria, only maternal mitochondria are passed on to the offspring. This organelle plays a central role in energy production. It functions like a digestive system at the cellular level, breaking down nutrients into usable energy. This energy powers nearly every activity in our daily lives, from walking and thinking to the simple act of breathing while asleep. The process of energy production in mitochondria occurs in 3 stages, yielding a total of 36 molecules of energy, known as adenosine diphosphate or ADP. The first stage produces 4 molecules, the second 6, and the final stage generates 24. It is for this reason that mitochondria are often referred to as the cell's powerhouses. Lysosomes are tiny, membrane-bound vesicles found in the outer fluid of the cell and are associated with the Golgi apparatus. They contain acidic substances that break down harmful bacteria and viruses that invade the cell. Lysosomes also assist in digesting nutrients such as proteins and fats, and they recycle worn-out mitochondria to maintain cellular efficiency. The cytoskeleton, of which there are thousands in every living cell, is principally responsible for protein synthesis. When a cell requires protein, mRNA delivers the genetic blueprint from the nucleus to the cytoskeleton located outside it. The cytoskeleton has two parts, a smaller section that reads the mRNA's message, and a larger section that constructs proteins from amino acids delivered by transfer RNA, or tRNA. This process forms a continuous chain of amino acids, assembling proteins according to the original instructions carried by the mRNA. The organelles mentioned here represent only a selection of the many intricate components found within the cell. There are, of course, many more to be discovered, though for those one must consult other, possibly less soothing sources. Cell division, or more precisely, cell duplication, occurs in two distinct forms, mitosis and meiosis. Mitosis is the process responsible for restoring and replacing damaged cells, such as those affected during bodily infections. It begins when the nucleus and chromosomes of a diploid cell, that is, a cell containing paired chromosomes, separate into two parts. The original cell then divides to form two genetically identical cells, each with the same number of chromosomes as the original. This process ensures that body cells, when replicated, maintain genetic consistency. Meiosis, on the other hand, occurs only in sexually reproducing organisms. This type of cell division leads to the formation of 4 reproductive cells, or gametes, from a single diploid cell. Meiosis proceeds in 2 phases. In the first phase, the diploid cell divides to produce 2 new cells with the same number of chromosomes as the original. In the second phase, these 2 cells divide once again, resulting in 4 cells. These final cells, sperm in males and eggs in females, contain half the number of chromosomes as the original diploid cell. The distinction between the 2 forms of division lies in the behavior of chromatids, which are the arms of a chromosome. During mitosis, these chromatids do not separate into individual divisions. However, during meiosis, the chromatids do split, allowing for the reduction of chromosome number and the eventual formation of 4 genetically unique cells. To move from cell division into the broader realm of genetics, we must now consider the contributions of Gregor Mendel and his foundational theories. The genetic theory begins with the understanding that one of the core characteristics of life is reproduction. Yet this reproduction is not random. A pine tree does not give rise to an oak, nor does a rhinoceros produce a giraffe. Offspring are invariably more similar to their parents than to unrelated members of the same species. This fundamental truth forms the basis of what is known as descent with modification. Even as traits are passed faithfully from one generation to the next, variations arise. These differences, subtle or striking, are what we call genetic variation. Human societies have understood and utilized these principles of heredity and variation for thousands of years in the selective breeding of plants and animals. However, the underlying mechanisms behind inheritance and variation began to be understood only with the advent of modern genetics in the 20th century. At the center of this development was Gregor Mendel, a figure often credited with establishing the basic principles of heredity. Born in 1822 to a German-speaking family in Austria, Mendel pursued studies in philosophy, science, and mathematics before becoming a monk. He later continued his education at the University of Vienna, where he focused on plant biology and mathematics. In 1857, Mendel began a series of experiments breeding garden peas in the monastery's Abbey garden. His purpose was to investigate how traits were inherited. He chose peas for their accessibility and diversity. For instance, one variety might have purple flowers, while another bore white ones. In the language of genetics, a character refers to a heritable feature, such as flower color, that varies among individuals. Each specific manifestation of a character, such as purple or white flowers, is referred to as a trait. Through these meticulous experiments, Mendel laid the groundwork for our understanding of how genetic information is transmitted, providing the foundation for modern genetic theory. The theory of growth and development can be organized into 10 foundational themes. The first of these is an examination of the nature of growth, reproduction, and developmental biology. At its core, developmental science seeks to understand how a living organism is formed, how it grows, and how it ultimately reproduces. Modern interpretations of this science emphasize that all these processes are governed by genes. These include the development and differentiation of cells, the assembly of tissues and organs, and the complex choreography of reproduction and maturation. To illustrate this, we move to the topic of sperm and egg, more specifically, the formation and development of the human body. Here, one might pause and ask, how does the fertilization of a single microscopic egg by a sperm eventually produce the astonishing complexity of the human form? What mechanisms orchestrate and regulate this transformation? And which elements contribute to the assembly of the body's organ systems? Central to this process is the DNA housed within the nuclei of cells. DNA acts as the master regulator, directing changes in the nature and function of each cell. One could think of it as a vast internal library, storing the blueprints for all cellular and developmental operations. Yet to make use of this stored information, the cell must rely on proteins. Proteins execute the functional tasks both inside and outside the cell. This naturally invites another question. Which proteins are responsible for which functions? And how is this specificity determined? Further inquiries lead us to the origins of sperm and egg cells. In males and females, these reproductive cells are produced in the testes and ovaries, respectively. Egg cells, in particular, begin their development early. By the time a female child is born, she already possesses approximately 400,000 egg cells, each paused at a preliminary stage of development. It is not until around the age of 12, during puberty, that these cells begin to progress beyond the primary oocyte stage. This maturation is triggered by signals from the brain, specifically the pituitary gland, a structure linked with the amygdala, releases 2 hormones directly into the bloodstream. These hormones travel to the gonads, where they act as catalysts, initiating the maturation of the egg cell. Once sexual maturity is reached, this hormonal regulation continues in the form of the menstrual cycle. Month after month, an egg matures and is then either fertilized or discarded, following a tightly controlled schedule of growth and decay. The male human body, too, engages in the biological theater of gamete production, specifically the formation of sperm within the testes. The male reproductive system comprises several parts: the testes, urethra, vas deferens, prostate gland, seminal vesicle, and penis. Each day, the testes produce around 200 million sperm cells. This prolific output is not arbitrary. It is triggered by hormones released by the pituitary gland, which is regulated by genes and situated in the amygdala region of the brain. These hormones, circulating through the bloodstream, not only initiate the production of sperm, but also aid in their maturation. Once matured, the sperm are transported into the epididymis, where they receive sustenance and are stored. In the continuation of this intricate process, the formation of an embryo begins once sperm reaches the female reproductive tract. Following ejaculation, during which approximately 300 million sperm are released, these cells can survive in the female reproductive environment for 4 to 5 days. Yet, despite this overwhelming number, only several hundred manage to reach the egg. The acidic environment of the reproductive tract proves lethal to most. Of those that do arrive, only one sperm manages to enter the egg. It does so by releasing acid from its acrosome, the tip of the sperm, which perforates the egg's outer membrane. This action simultaneously renders the membrane impermeable to any additional sperm, preserving the integrity of the genetic material within. The successful fusion of a single sperm with an egg forms a zygote. This cell then divides into 2 smaller, genetically identical cells. At this stage, the developing mass is referred to as a morula. The morula continues its division as it travels, eventually becoming a structure called a trophoblast, which envelops an internal cluster of cells known as the embryoblast. Fluid accumulates between these cell groups, resulting in a new formation called a blastocyst. From this point, the inner cells begin the transformation into embryonic stem cells, capable of becoming any cell type. These versatile cells migrate to various parts of the developing embryo, preparing to shape future tissues and organs. For these embryonic cells to transition into specialized states, they undergo a process called cellular differentiation. This is the method by which a single general cell develops into a specific type, be it a blood cell, skin cell, or organ cell. Collections of similar cells form tissues, and in turn, distinct tissues assemble into organs. This precise choreography allows the fetus to develop its organ systems efficiently. However, should cellular differentiation falter, developmental abnormalities may arise. This naturally invites several pressing questions. What directs this gradual cellular transformation? How does one cell type become another? How are these cells organized into functional tissues and organs? The answer lies in cellular interactions. When cells come into contact, their genes are activated, prompting the production of various proteins. These proteins are the agents of differentiation. Growth factor proteins facilitate signal transduction, communicating with surrounding cells, while transcription factors ensure that the correct genes are expressed. These proteins influence both internal and external cellular environments as they perform their designated roles. As embryogenesis proceeds, the structural layout of the embryo undergoes significant reorganization, beginning with gastrulation. During this process, 3 primary germ layers emerge: the endoderm, mesoderm, and ectoderm. The endoderm, forming the innermost layer, gives rise to internal organs, including the heart, lungs, liver, spleen, kidneys, stomach, colon, intestines, bile system, urinary bladder, and gonads. The ectoderm, the outermost layer, contributes to the development of the skin, the nervous system, and pigment-producing melanocytes. The mesoderm, nestled between the two, is responsible for producing muscles, bones, and components of the circulatory system. From a single cell, an entire organism begins to take shape, but not without the guiding hand of cell signaling. This process begins when a cell receives cues from its environment or neighboring cells. These cues initiate a series of intracellular events known as signal transduction, enabling the signal to cross the cell membrane. While some chemical signals bypass the membrane entirely, the majority interact with specific receptors on the cell's surface. These receptor proteins serve as gatekeepers for communication, only binding to their designated signal molecules. Once inside, the signal is passed along a relay of proteins, each modifying the next in the sequence. This cascade, known as the signaling pathway, directs the signal toward a specific destination, whether that be the cell's nucleus, the membrane, or other compartments. The cell, in essence, becomes a bustling information hub, ensuring that messages are received, interpreted, and acted upon with cellular precision. There are various types of signaling pathways within the cell, and some of these fan out in multiple directions, dispatching signals to different destinations inside the cell. These signals may intensify as they pass from one protein to the next, or they may weaken during events such as cell division. Regardless of the trajectory, the signal invariably provokes a significant cellular response. Once a signal arrives at its intended destination, it alters the behavior of the cell. This behavioral change depends entirely on which proteins participate and perhaps interfere in the process of signal transduction. To understand reproduction, biologists have turned to a variety of animal models. These include organisms that are relatively simple and readily accessible, such as mice, bees, frogs, chickens, and certain aquatic insects. By studying embryonic development in these species, scientists can observe how proteins and genes function, how they influence the formation and regulation of organ systems, and how these mechanisms might translate to human development. The process of embryogenesis, the sequential development of the embryo within the womb, reveals notable changes at each week. By the 5th week, the embryo begins to develop the brain, spinal cord, and heart, along with early structures of the stomach and small intestine. In the 6th and 7th weeks, the bones of the limbs start forming, and the brain divides into 5 parts. Some of these regions begin to generate nerves and tissues that will eventually form the eyes, ears, vertebrae, and additional bones. The heart grows more complex and starts to beat, initiating blood flow through the fetus's primary blood vessels. By the 8th week, the ends of the limbs, fingers and toes, become more defined, and the lungs begin their formation. In the 9th week, nerve cells for hair growth emerge, limbs become more prominent, and key organs begin their development. By the 10th week, eyelids and the outer ears start to form. This also marks the end of the embryonic period. Between the 11th and 14th weeks, facial features become more detailed, limbs lengthen, and the reproductive system begins to form. Red blood cells start developing in the lungs. At this stage, the baby's head constitutes nearly half of its total body weight. From this point until the 28th week, the baby's eyes remain closed. During weeks 15 through 18, the skin remains transparent while hair begins growing on the head. The mouth gains the ability to move, allowing for sucking and inhaling. The liver and gallbladder begin producing fluids. Muscles form, bones strengthen, and the baby starts to move. By the 19th week, the fetus is capable of hearing. At the 22nd week, the fetus develops a fine layer of hair on the head, eyelashes, and eyelids. Fingernails also begin forming. As muscles develop further, fetal movements become stronger and perceptible to the mother. A stethoscope can now detect the fetus's rapid heartbeat. Between weeks 23 and 25, the bone marrow starts producing blood stem cells. Although parts of the lungs form, the fetus cannot yet breathe due to the incomplete development of the airways. At week 26, eyebrows, eyelashes, and the full structure of the eyes develop, along with the palms, soles, and the diaphragm encasing the lungs. From weeks 27 to 30, there is accelerated development of the brain and nervous system, which now begins to regulate the body's activity. The fetus can open and close its eyes, and the respiratory system continues to mature. Between weeks 31 and 33, fatty tissue begins to develop in the lungs, allowing for the initiation of breathing, though the lungs remain incomplete. The skeletal system is fully formed, but remains soft and pliable. The fetus starts to accumulate iron, calcium, and phosphate. By the 38th week, fat continues to accumulate, and the fetal body begins to shed its fine hair. From weeks 39 to 42, hair remains only on the upper parts of the hands and shoulders, while the scalp hair thickens and the nails lengthen. In alignment with the mother's physiology, birth typically occurs between the 42nd and 44th week. Turning to the menstrual cycle, it typically spans 28 days, though this can vary. Middle-aged women tend to experience cycles lasting between 21 and 35 days, while for teenagers, cycles may range from 21 to 45 days. Day 1 of menstrual bleeding marks the beginning of the cycle. Following this, the ovarian follicle, which houses the oocyte, begins to mature. By the end of menstruation, levels of developmental catalysts wane, but follicular growth continues under the influence of enzymes. Oocytes within the ovarian follicle are then activated. Follicular development usually continues for 7 or 8 days post-menstruation. Around day 14, one dominant follicle outpaces the others in size and development. At this time, the endometrial lining thickens, preparing for potential implantation. This lining becomes enriched with blood and nutrients. On the 14th day, the mature egg is released from the ovary, a process known as ovulation, triggered by enzymes that rupture the follicle and allow the egg to enter the fallopian tube. If the egg encounters sperm within the next few days, fertilization may occur, and the resulting zygote continues to travel until it attaches to the uterine wall. Fertility is thus highest between days 11 and 18 of the menstrual cycle. If fertilization does not take place, ovulation ceases by day 25. The unfertilized egg then degenerates and is expelled through the fallopian tube. Finally, a word on the virtue of multicellularity. Organisms may be unicellular or multicellular, and while unicellular organisms are more numerous, multicellular organisms benefit from a division of labor. This cellular specialization allows for more efficient functioning, and it is this efficiency that has driven the evolutionary success and diversity of multicellular life. The benefits of being multicellular are rooted in the concept of division of labor. In multicellular organisms, each cell assumes a specific specialized role. These responsibilities are non-transferable. A cell cannot adopt the function of another. The nucleus will not perform the role of the cell membrane, and the membrane will certainly not attempt to manage the tasks of the nucleus. This specialization is crucial for the organism's function and survival. Different cell types exist because specific functions must be carried out by specific tissues. For instance, cells destined to become lung tissue are not capable of forming the tissues of the heart or brain. Each tissue, therefore, has its own structure and form, tailored to its unique function. Neurons, for example, are long and slender, perfectly suited to transmit electrical impulses across the body. Skin cells, in contrast, are flat and densely packed, designed to form a protective barrier against the external environment. This elaborate division of function means that the body can carry out a wide range of complex activities. A collection of similar cells forms a tissue. Various tissues come together to construct an organ. Stem cells, once influenced by chemical signals, irreversibly specialize into distinct cell types. These specialized cells then build each organ, laying the foundation for the intricate architecture of the body. All cells in the body contain the same DNA, yet they exhibit different characteristics. This is due to selective gene expression. Although the genetic material is identical, different genes are activated at different times based on the cell's role. A skin cell, a blood cell, and a muscle cell all originate from a single fertilized egg, yet through precise regulation of gene expression, they differentiate into over 3 trillion uniquely functioning units within the human body. As these cells specialize and aggregate into tissues, and those tissues organize into organs, the organs themselves coordinate to form organ systems. Each organ completes a distinct task. The nervous system, for instance, receives signals from across the body and transmits them to the brain, which then responds appropriately. The circulatory system distributes oxygen and nutrients, while also ferrying waste to the kidneys. The immune system protects the body from external threats, and the reproductive system enables the continuation of the species. Every muscle movement, from a blink to a backflip, is orchestrated by the muscular system. Together, these systems operate in harmony, making life possible. Now, while this complexity is usually a triumph of coordination, it is not immune to error. During embryogenesis, gene mutations can lead to abnormalities, twins conjoined at the chest, missing limbs, congenital blindness, or other developmental anomalies. To understand how these arise, we turn to the theory of gene mutation. Gene mutations are alterations in the DNA sequence of a gene and fall into 2 primary categories. Hereditary mutations are passed from parent to child via the germ line, sperm, or egg, and are therefore present in every cell of the offspring's body. These are lifelong and systemic. On the other hand, acquired or somatic mutations occur at some point during a person's life. They arise in individual cells and do not affect every cell in the body. Environmental factors such as UV radiation or random errors during DNA replication can cause these mutations. Importantly, somatic mutations are not inheritable. Mutations can disrupt the production or structure of proteins, resulting in cells with abnormal or dysfunctional behavior. These changes manifest physically during development. In model organisms, such as flies, 3 general mutation types have been observed. A deletion mutation may result in the loss of a body part. A point or missense mutation causes more controlled, targeted faults. A frameshift mutation, a particularly disruptive kind, can alter entire sections of a body region, often with dramatic effects. In summary, multicellularity affords complexity, specialization, and resilience. But with these advantages comes the risk of intricate failure. A single faulty gene can ripple through a system of astonishing precision, sometimes leaving a permanent mark. All living organisms, and especially animals, face a constant threat from external viruses and germs. In addition, damaged or unhealthy body cells may pose a risk of becoming infectious themselves. To manage these threats, the body is equipped with 3 distinct layers of defense. The first consists of the skin, membranes, and various secretions, broad and impartial protectors, indifferent to the identity of their assailants. The second line of defense includes white blood cells, defensive proteins, and the inflammatory response. This too is nonspecific. Activated by chemical signals, phagocytes and small protein defenders converge on intruders that manage to bypass the first barrier. A hallmark of this defense is inflammation, a sign the second layer has sprung into action. The third layer of defense is the immune system proper, a network of cells, tissues, and organs with a particular set of skills. This layer delivers a specific immune response, customizing its attack to the nature of the invader, whether virus, bacteria, parasite, or fungus. The immune system can identify and remember millions of different threats and respond with precision. Its critical function lies in distinguishing between what belongs and what does not, between self and non-self. Cells marked as foreign elicit immediate retaliation, and anything that can trigger this response is termed an antigen. Antigens include not only microbes, but also foreign tissue, explaining, for instance, why organ transplants may be rejected. Occasionally, the system misidentifies its own tissues as threats, resulting in autoimmune diseases such as certain forms of arthritis or diabetes. The immune system's organs, known as lymphoid organs, are distributed throughout the body and host lymphocytes, small white blood cells central to immune function. All blood cells originate in the bone marrow, including immune cells. The thymus, located behind the breastbone, is where T lymphocytes, or T cells, undergo maturation. Lymphocytes travel through both blood vessels and a parallel system of lymphatic vessels. These vessels, filled with a clear fluid called lymph, allow cells and fluids to circulate between the blood and lymphatic systems. Along this network are lymph nodes, small, bean-shaped structures stationed in areas such as the neck, armpits, abdomen, and groin. Within these nodes, immune cells gather and interact with antigens. Foreign particles and immune cells enter lymph nodes either through lymphatic channels or tiny blood vessels. Once they exit, lymphocytes rejoin the bloodstream and circulate through the tissues, continuing their patrol for antigens. After a time, they re-enter the lymphatic system, perpetuating the surveillance cycle. The spleen, nestled in the upper left abdomen, serves a similar role. It houses immune cells and facilitates their interactions with antigens in specialized compartments. The immune system's armory includes more than lymphocytes. It also contains phagocytes, cells that engulf and destroy invaders, and others that work in cooperation. Some immune cells communicate directly through contact, others by releasing chemical messengers. Though the immune system keeps only a small number of each type of immune cell on standby, this lean force can multiply rapidly when activated by a matching antigen. Once the threat is neutralized, most of these cells disappear, leaving behind a few sentries to stand watch for any future incursion. All immune cells begin as immature stem cells in the bone marrow. In response to signals such as cytokines, they develop into specific cell types, T cells, B cells, or phagocytes. B lymphocytes, or B cells, operate primarily by producing antibodies, which are secreted into bodily fluids and seek out antigens. These antibodies can bind antigens in the bloodstream, but cannot penetrate infected cells. That job falls to the T cells and other immune cells. Each B cell is programmed to make one type of antibody. One might target the cold virus. Another might specialize in pneumonia-causing bacteria. When a B cell encounters its specific antigen, it produces plasma cells, which are essentially antibody factories, churning out millions of identical antibodies. The interaction between antibody and antigen is much like a key fitting into a lock, marking the invader for destruction. T cells, unlike their B cell counterparts, are not interested in free-floating antigens. They detect antigen fragments displayed on the surfaces of infected or abnormal cells. There are two primary roles for T cells. Some direct and regulate the immune response, while others attack infected or cancerous cells. Helper T cells coordinate the defense effort, stimulating B cells, summoning phagocytes, and activating other T cells. Killer T cells, or cytotoxic T lymphocytes, attack infected cells directly. These are especially valuable in the fight against viruses, which tend to hide within host cells. Killer T cells recognize viral fragments peeking through the membrane and promptly eliminate the compromised cell. Natural killer, NK cells, another variety of white blood cell, are similarly equipped to destroy. Armed with granules containing lethal chemicals, NK cells can recognize and attack a broad array of foreign cells. Though they operate with less precision than killer T cells, both are lethal on contact. They bind their targets, aim, and release a fatal volley of chemicals. Phagocytes are large white blood cells designed to ingest and digest microbes, along with other foreign particles. Among these are monocytes, which circulate through the bloodstream. When monocytes enter body tissues, they transform into macrophages. These specialized macrophages can be found embedded throughout various organs, the lungs, kidneys, brain, and liver among them. Macrophages are multi-purpose operators. They function as scavengers, clearing out worn-out cells and debris. They also present fragments of foreign antigens in a way that attracts specific lymphocytes and, quite industriously, release a wide range of chemical signals known as monokines, essential messengers in the immune response. Another group of immune cells, the granulocytes, are named for their granule-filled interiors. These granules are loaded with chemicals that destroy microorganisms. Some of these substances, like histamine, also contribute to the familiar inflammation and allergic reactions. Neutrophils, a type of granulocyte, are also phagocytes. They use their chemical stockpiles to break down what they ingest. Eosinophils and basophils, while not phagocytic, release their granules onto surrounding harmful invaders. Even platelets, typically associated with blood clotting and wound healing, contribute by activating elements of the immune response through their granules. Communication within the immune system is coordinated by cytokines, protein messengers secreted by cells to influence the behavior of other cells. These signals help control immune cell growth, activate responses, and draw cells to infection sites. Cytokines essentially serve as the immune system's internal postal service, directing troops where they are most needed. The concept of immunity itself comes in two broad flavors: natural and acquired. Long before microbes were visible, people noticed that survivors of certain diseases rarely suffered them twice. This acquired immunity is due to memory cells, T and B cells that remain in the body after an initial infection. Upon re-encounter with the same antigen, these cells rapidly respond. The strength and longevity of immunity vary depending on the nature of the antigen, its quantity, and its entry method. Some individuals respond to a given antigen with vigor, others weakly, and some not at all, often due to inherited genetic differences. Immunity can be sparked through infection or through vaccination. Vaccines are weakened or inactive versions of pathogens, designed to provoke an immune response without causing full disease. Immunity can also be borrowed temporarily via injections of antibody-rich serum called antiserum. This passive immunity is short-lived but useful, such as when protecting travelers from hepatitis A. Infants begin life with immature immune systems, but benefit from maternal antibodies, both from the placenta and for the breastfed from the milk, particularly for gastrointestinal protection. Despite its strengths, the immune system is not infallible and may suffer from various disorders. One common type is allergy. Allergic diseases arise when the immune system overreacts to benign substances, such as pollen or dust, interpreting them as threats. This hypersensitivity involves IgE antibodies, each tuned to a specific allergen, oak pollen, ragweed, and so on. Autoimmune diseases occur when the immune system mistakenly identifies the body's own tissues as foreign. In these cases, T cells and autoantibodies attack healthy cells. For example, in diabetes, T cells destroy insulin-producing pancreatic cells. In rheumatoid arthritis, an autoantibody called rheumatoid factor is commonly found. Lupus, or systemic lupus erythematosus, SLE, involves antibodies that target multiple types of the body's own cells. The causes of autoimmune diseases remain elusive, but are thought to involve a mixture of environmental and genetic factors. Triggers might include infections, sunlight, certain drugs, or hormonal shifts, particularly as many autoimmune disorders are more prevalent in women. Immunodeficiency disorders occur when components of the immune system are absent or dysfunctional. These can be inherited, result from infections, or be induced unintentionally by treatments like chemotherapy or immunosuppressants used after transplants. Temporary immune suppression can follow common viral infections like flu or mononucleosis, and may also arise from surgery, malnutrition, stress, or smoking. Some children are born with incomplete immune systems, lacking B cells, T cells, or in rare cases, all major defenses. One severe acquired immunodeficiency is AIDS, caused by the HIV virus. HIV disables critical T cells and can hide in them for extended periods, eventually undermining immune function and leading to opportunistic infections and cancers. The immune system itself can become the target of malignancy. Leukemias are cancers of white blood cells, and when plasma cells grow uncontrollably, the result is multiple myeloma. Though scientists have uncovered much about immune function, many questions remain. With advancing technology, researchers are now able to observe immune cells like B and T cells as they interact within lymph nodes and other tissues. These new tools, coupled with expanding genetic knowledge, are opening new doors to understanding how our bodies defend against disease.